next, we're going to have our Director of Science, Dr. Hourinaz Behesti, come up and talk to all of you about the updates on science. It's going to include Sue's work and the other scientists that Sue mentioned and a few others. Hourinaz has been part of our foundation also since the beginning as a scientific advisory board member. But about a year and a half ago, she told me that she was leaving the university at Rockefeller University to pursue some other things. And we luckily were able to convince her to also become our Director of Science. And over the last year, she has really elevated all of our work to the next level and brought us much closer to getting to a clinical trial. So really so grateful for Hourinaz and excited for her talk. And just so you know, we will have Q&A after. We're going to have a panel discussion with Hourinaz, with Sue. Shibi will join us, and I'd love, since Corinne and Fabian have been so kind to stay, we'd love to have you guys come up for the panel as well.
Okay, thank you, Nasha. It's really a great honor to be here amongst you all. As Nasha mentioned already, I'm a scientist. I'm a developmental neurobiologist with a long-standing interest in human brain development and really what makes us uniquely human. And it's really been transformative to get involved with the foundation and really see the patient side of things and get to interact with you all. And I can probably speak for all the scientists who are here in the audience who spoke on Thursdays, whose work I'm going to try and do justice by sharing with you, that we just by talking to you, we get to learn so much more that we can read in papers. And so this dialogue is really important. So today I'm going to summarize our efforts, sort of give you an overall overview of what we are doing, and also talk about what is known about FOXG1 biology in the field in general, and also the progress that has been made in the past couple of years, including the fantastic talks that we got to hear about on Thursday. So I'm going to start off by just also trying to put things in context for you guys a little bit. So what we are going after initially are sort of therapies that we call more possible therapies. These are things that are already being given to other patients in the clinic. So they've kind of gone through the regulatory processes. It's already been approved. So the barrier of getting it to patients is actually is sort of lower, and it's going to be faster. And then I'm going to talk a little bit about just what we know about FOXG1 biology, and sort of show you some images to kind of try and put what scientists talk about into context for you, and then finish off by telling you about the new advances in the field, much of which has been funded by the foundation. So we already heard about the drug development process from Alison briefly this morning. There are lots of different steps involved, and it's it can be a long process. Oh, I can't. Oh, there we go. So it starts with a discovery phase. So what is a discovery phase? It's basically the phase where you go in and try and understand as much as possible about the biology of the gene and of the syndrome itself. And so to be able to do this, we have to create models, animal models, which is what Sue was talking about. And so these include mouse models, they include cells, human cells that we get from you guys in terms of getting fibroblasts that we can then turn into nerve cells. I'll talk to you guys about it a little bit more in the next few slides, how we do that, so that we can actually really try and understand the biology of the syndrome and the gene. And then in this phase, we're hoping to really hone in on a target, something that we know if we go after it, we'll be able to improve this condition. And so once we have that, then we enter the preclinical phase, which is the phase where we basically try to adapt different methods to actually deliver a therapeutic and see how much correction we get. And that's usually done in the animal models and the cell models that we create. And it's also at this stage that we try to also assess how safe the drug is that we have now developed and we're giving to the animals. And once it's looking good at this stage, then we can enter the human phase, which is clinical trials. And there are 3 main phases to clinical trials as well. The first phase is just assessing the safety of the drug. And then after that comes the same thing as what we did in the animal models, which is to see, does this drug actually improve anything? Does it actually fix any of the symptoms? And then it goes to the FDA, and the FDA basically will make a decision of whether they will allow this drug to actually be given out to patients in the population. And so to be able to really convince the FDA, which is what our end goal is, we really need to do all these steps really properly with really sort of like rigorous data and rigorous analysis and dissemination of the information to really be able to convince them at this end stage. So as you can imagine, this is quite hard. It's lots of different steps. And unfortunately, there are many promising drugs, so things that actually look like they could work. They go all the way to clinical trials, and then they fail. And they fail for silly reasons. They fail because they weren't given to the right cells that need to receive them in the body. Not because they don't fail because they're not working, they just weren't delivered in the right place. Or they fail because we didn't spend enough time to understand the biology better. And so again, we delivered in a way that wasn't appropriate. Or that we weren't able to really measure you heard about outcome measures. So the things that we're looking to fix, we weren't really able to measure that in a proper way to be able to actually convince the FDA that we were able to fix something. So these are all the things that we are working hard to kind of do before we get to clinical trials, so that the drugs that we bring there are not being approved because of these reasons. And so we aim to find the most effective therapies and do the work needed prior to trials so that they actually succeed in trials. So these are 3 examples of technologies, methods that are currently in the clinic, meaning that they've already been given to other patients with other kind of symptoms. So you may have heard of gene therapy, which is sort of a way of replacing the gene from outside. You heard ASOs already mentioned today, and you heard about small molecules. And I'll tell you a little bit more about how each of these sort of work and how they might be different from each other. But the strategy of the foundation right now is basically doing this early work that I described, how important it is. So the first step is the discovery stage, and we are very much still in this discovery stage. And that includes basically building models and tools, which Sue's lab is doing. We have Fabienne's lab, who's also here, who's doing that. We have Corinne's lab doing that. The next step is and also creating sort of other tools and methodologies to really be able to measure things properly and show that we can measure things properly. And we are basically trying to find viable targets. So like where in the cell can we go in and deliver a drug so that we can actually fix the way that the cell is behaving. We are also focusing on trying to understand biomarkers. You may have heard about biomarkers. It's really a critical sort of part of the journey before getting to clinical trials, and that's actually one way that you guys all can help. We can talk about that later. And we're testing different methods that are available that I mentioned, and these are sort of referred to as targeted approaches because we already know the target. The target is FOXG1. So we have ways of going in and manipulating FOXG1, and that basically falls under ASO, gene therapy, CRISPR-AI, RNAi methods. And then we're also undertaking what's called more of a blind approach. So we don't know the target, but we know that the cells behave differently from FOXG1 patients compared to control patients. And so without really trying to understand the underlying mechanisms, we just go in and try and make it look normal again. And we can do that by doing these sort of, it was called high throughput screenings, where we test thousands of drugs and then just see which one seems to work. So that's sort of like a fishing method more. And so by the end of this discovery phase, we are hoping to have identified a viable target that we can pursue. We are hoping to have identified sort of, or understood better, what is the timing? When do we need to get in there to deliver the therapy? We are hoping to have identified exactly where in the body we need to go and what cell types we need to target. And we're also hoping to have identified or really demonstrated that we can measure changes in a meaningful way and do them consistently in animal models in a way that it relates to what it looks like in patients. And then the next phase will be to try and test the safety. So is the drug safe enough to give to kids? Is it causing any sort of toxicity? And then the next stage will be to enter clinical trials. So I just thought I'll bring it down really to the basics first before we really jump in. So what is FOXG1? It's a gene, as you all know. It's called a transcription factor. And transcription factors are a category of genes that their sort of sole function is to switch on or off other genes in the genome. So your genes are encoded by DNA. And DNA does, we have so much genetic material in all our cells, and DNA kind of just needs to fit into our cells. And the way that evolution has sort of devised a way of doing that is by packaging DNA really tightly. So this is sort of like the first order packaging of DNA, which we refer to as chromatin. And then this gets packaged even more into this structure, which is called chromosomes. And I'm sure you've heard of chromosomes before, FOXG1 being on chromosome 14. And, but really what is sort of key to biology are proteins, okay? So it's really all about proteins. They're really the building blocks of cells. And where do proteins come from? They come from DNA. So if you think about DNA as the software, it's sort of like the blueprint. It holds all the information about what your cell needs to, what proteins your cell needs to make. DNA gets made into RNA as a sort of middle step. And then this RNA basically is the building block for proteins. So once you have the RNA in cells, then the cell knows, okay, these are the proteins that I need to make. And it goes on to form proteins, which actually carry out all the functions in the cell. So what does FOXG1 do? So basically, before Thursday, we knew that it's a transcription factor. After Thursday, we found out that it actually does lots of different things. So it binds to chromatin, this sort of higher order structure of DNA. It is a transcription factor. We already knew that. It got confirmed. And it is regulating how proteins are made. So it's really acting along this whole pathway. And this is obviously very interesting from a biological point of view, but it also opens up possibilities for therapeutic interventions. You can go in at multiple steps. And sorry about the resolution of this. I basically just copy and pasted this. I wanted to include this because I just thought it would be fun for you just to see where the gene is present in the body. So this is a cross-section of a mouse embryo. This is a very, very early stage embryo. This is the head. This is the tail. This is sort of the trunk. And this is the brain area, all of this. And what you see in blue is where FOXG1 is present. So it's really, what's really remarkable about this gene is that it's very localized to just the 4 brain structures of the brain. And these are really the structures that are thought, or classically thought, to underlie higher cognitive functions. So they're also giving rise to sort of motor control, but are also really controlling a lot of cognition, problem solving, decision making, etc. And FOXG1 continues to be expressed after the embryo is born, after the fetus is born, postnatally. And you can see again that it's expressed sort of it's very localized to just the front parts of the brain. So our aim is obviously to develop therapies, and to be able to really test these therapies, as I mentioned, we need to develop tools. So for the past few years, we have been very busy, the scientists have been very busy in creating mouse lines, fish models. So we have currently 7 that are being studied and being tested. We've also created human cell lines from material that probably most of you have donated. So we currently have 54 cell lines that have been made and are being analyzed. And as you heard earlier, there's also been a lot of activity in trying to understand patient data through the natural history process, natural history data, and we have 100 of them completed. And so before we go on to testings in these models, we want to first understand how well these models, thank you, how well these models are sort of showing what we see in patients, right? So one of the key things that we see in a lot of the patients is that you may have heard of as well is that the corpus callosum is not normal. So what is the corpus callosum? It's this part of the brain here that connects the right hemisphere to the left hemisphere. It's sort of like a fiber track. Other things that we consistently see in patients is microcephaly. So what we want to see in the models is whether we can actually recreate that. Do they have smaller brains? Another thing that we consistently see is that more at the cellular level, we see sort of changes in the composition of the cells in the brain. And so that's another thing that scientists are looking at very carefully to try and understand and identify in these models. So now I'll tell you more about what we have found out about the models and sort of the characteristics of the models. So as I mentioned, we are making induced pluripotent stem cells from patient samples and from fibroblasts that have been donated. So you can take a piece of skin from a patient, and by giving it a cocktail of factors that actually this was developed and really discovered in 2006, and people in Japan actually got the Nobel Prize for this. So it really revolutionized the field in being able to do this, be able to actually study patient cells directly in a dish. So this is what we're doing here. We take a piece of skin, we can turn them into stem cells, so like very primitive cells, and then we can direct them to any type of cell, other cell of the body. So from a skin cell, we can actually now create nerve cells, and we can actually study the nerve cells of patients directly in a dish. And so we have several projects on this. I'll mention one of them who have really spearheaded this work. They're actually in the audience here, Angels and Tiago sitting at the back there from the Mucci lab at UCSD. So they have reprogrammed 6 lines to make iPSCs, and then they've gone on to create these mini brains. So they sort of like these aggregates that are of these cells that they are giving certain factors to turn them into sort of a mini brain. It doesn't completely look like a human brain, but it's the closest thing that we have right now to be able to actually sort of draw some conclusions about what is happening in the human brain. And so they're able to study them and see how well they correlate to what's happening in the human brain during development. So they can study them longitudinally. They can look at things like electrical activity in these cells, which is what is depicted here. And they have previously published papers showing that this actually correlates really well with EEG findings, so we can correlate that back to what is being measured in patients. And you heard briefly from Sue and her work on the mouse models. What they have found is that they are all slightly different from each other, and that is a sort of positive thing to have found, because the children are all different from one another. There is really a spectrum of sort of symptoms in the children as well. And she's finding that the structure of the brains are not developing properly as seen in the children. She's testing a whole bunch of behavior tests that are sort of like proxy for motor function and cognitive functions in humans. And she shared data about sort of abnormalities in these behavioral tests. We got to hear from her previous postdoc about the gastrointestinal issues. So there's a number of these mouse models are actually showing constipation. And we're also planning a new study. We're launching a new study in collaboration between Sue and Jean Paz's lab, where we're going to really deeply characterize the seizure abnormalities in these models and see how well they correlate with patient population. So here I'm going to tell you about one of these sort of behavioral tests that we're doing in mice. This is the work of Goichi Miyoshi from Japan, who's a scientist that we are funding. And this is also one of my favorite tests. People on the board who get to hear me talk about this, they've heard about this many times. So this is a test of sociability in mice. You'd be surprised they can be social too, like humans. It's called a three-chamber social assay. It has basically three chambers that are connected. So you can put a mouse here, and the mouse is allowed to roam freely between these three compartments. And so what you can do in terms of in trying to understand sociability in mice is that you can put a mouse here in the middle, and then you can put another mouse here in this left chamber, and then you can measure whether the mouse prefers to go and say hi to this other mouse, or if it prefers to go to the other side and not bother with the mouse. So you can see that here, and then these squiggly sort of purple lines are tracking the movement of the mouse. So here in a normal mouse, he's been put in the middle, and as you can see here, he spends much more time saying hi to what we call a new mouse, instead of going and hiding in the empty chamber. However, in the FOXG1 model, you can see pretty much the opposite. So the FOXG1 mouse prefers the empty chamber compared to going and sniffing out the other mouse. And then if you add another layer to this, so you have the mouse, the original mouse here, you have the mouse that now it's been formed, that is kind of interacted with, so it kind of knows this mouse a little bit. And then you can put a new mouse in this chamber here. And then again, you can measure and see how this mouse is traveling. Does he prefer to go and hang out with the old mouse, or does he prefer to go and sniff out the new mouse? And as you can see here, again, a normal mouse prefers to go and explore the new mouse and get to know the new mouse over hanging out with the old mouse. But in the FOXG1 situation, the mouse actually prefers to hang out in the middle, where the other two mice are not present. And so this is a way of really being able to test some behavioral deficits in these mouse models that we have, and I'll show you, I'll come back to this in the next few slides. But moving on to the zebrafish model, so this is a fish model. Surprisingly, fish have many similarities to the human brain, actually. And so one of the similarities that we see is the absence of the corpus callosum. So this is the equivalent of corpus callosum in fish that is missing in these fish models. And this is the work of Corinne, who's here at the back. And she's embarking on a number of different sort of projects to try and understand the basic biology of FOXG1. And one of the interesting things that she's finding, as she reported on Thursday, was that she actually sees different forms of the protein, of FOXG1 protein, which is really unexpected, because we thought if you have a mutation in one copy of the two genes, what you would expect is that you will just have half the amount of the protein of FOXG1. But what she's finding is that actually the cells end up producing a shorter form of FOXG1, and that is important to know, because that again is going to inform the type of therapeutic modality that we're going to pursue. So this is just to highlight that our research strategy is really being informed by what we are learning in the models and how important it is to really understand the models better. So back to this, my favorite. So this was really exciting for me to see as a developmental biologist, and also just for the field of FOXG1, because this is the first experiment that actually really demonstrates that you can correct symptoms in a FOXG1 model. And of course, this gives hopes that the same is going to be true in the human situation. So you're familiar with this. So the mouse prefers to go and hang out with a new mouse. So what Goichi in Japan did was that he transplanted a type of cells back into the brain of this FOXG1 model. And what he saw was that instead of going to the empty chamber, or instead of preferring to hang out in the empty chamber when he has two other mice next to him, is that he actually starts behaving much like the normal mouse, in that after the transplantation of these cells, we're able to rescue this sociability abnormality, and the mouse now actually goes and sniffs out the novel mouse much more than it prefers to be alone. So this really showed us the first sort of like evidence that we can indeed rescue something. So what is it that we're trying to rescue? So the most obvious thing to do for FOXG1 is to go after FOXG1 itself, is to correct FOXG1 levels itself. And so we believe, although some of the work that is coming out now is showing that it's a little bit more complicated than that, but in general, we know that we have, so if you can just imagine this bucket representing a cell, you can imagine that in the FOXG1 syndrome cells, the bucket is half full. And what we are trying to do is to increase the level of water back to sort of normal levels again. And there are different ways of doing this, and I talked about gene replacement therapy. So gene replacement therapy would be the equivalent of pouring water back into the bucket from the outside. ASO therapies, CRISPR-A, CRISPR-I, RNAi, all of these other therapies, tRNA therapy, which you heard briefly mentioned by Sue, involves increasing the levels of water from the inside. And so one of the therapeutic modalities that we are pursuing, so this is a work of Sue's lab together with Catherine Meyer's lab and Shibi, who's in the audience that you heard, is gene therapy. So this involves basically pouring water back into the bucket from the outside. And so the way we're doing that is by giving the cells a normal copy of FOXG1 using a virus. And so here they are showing how when you inject the virus, where it ends up in the brain. So first we can see that it's actually, so all the red parts is where the virus has gone in the brain. And luckily we see that it actually ends up going to all the areas that we want to try and hit in FOXG1. So all the sort of frontal areas that I was talking about that are involved in higher cognitive functions, we're able to hit those. And this is also really exciting data. Again, as a developmental biologist, like really amazing to be able to see this, and really sort of, I think, encouraging again for the field. So I talked about the corpus callosum and how the patients have defects in the corpus callosum. So this is the corpus callosum in mice. And in this FOXG1 mouse model, you can see that a bit of it is missing. And what they were able to show was that when they inject a virus containing a correct copy of FOXG1 into this mouse model, they're actually able to restore the corpus callosum. And so this is one of the, this is a really big deal. And so obviously this is one of the main sort of symptoms and of abnormality that we see in the patients and in the model. And so I won't go into too much detail, into too much data, but they're also seeing rescue in other parts of the brain, and they're also seeing improvements in a bunch of behavioral analysis that they're doing in the mice. And so the next step, so this is a great start, really encouraging, and we have something to work with. But we still need to improve it. We need to understand it a little bit better because we need to make sure that it really just goes to the correct parts of the brain and no other parts, because we want to avoid targeting regions that don't need FOXG1. And we also need to understand really what cells are being corrected and how that could be related back to patients. And of course, we need to understand how toxic it is before we can move on to the patient. So these are the next steps that are currently being undertaken. Another really cool approach that we got to hear about was from Fabian. So what he did in a different mouse model was that he actually identified a different target. So again, you know, how important it is to do this early discovery work. You can actually go at it from like different angles. So he identified another protein called PLP, which is extremely important for myelin. You might have heard about myelin in relation to if you know somebody who has MS or other demyelination disorder. So myelin is something that sort of wraps around nerve cells and allows nerve cells to send signals much faster. So it's very critical for functioning of the brain. And what he found was that this protein that is very important for formation of myelin is up in FOXG1 mutants, and he designed an ASO therapy to just bring the level of this protein down, and he was successful in doing that. He demonstrated that he was indeed able to do that, and then he did this behavior test where, so this is a grip strength test that we can do in mice. So he let the mouse hold on to this sort of barrel, and if you sort of pull the tail of the mouse, it will just like hold on until you pull too much, and it will eventually let go. And then you can measure the strength of like how much did I have to pull for this mouse to pull away. And this is something that is reduced in the FOXG1 mouse model, and he was able to see that just by decreasing the levels of PLP1, he was able to increase the grip strength in these mice, which is obviously very important for a lot of the sort of motor components of the FOXG1 children. And so these were the targeted approaches. I also briefly mentioned that we're doing this sort of like blind approach where we don't know what we're targeting, but we have a bunch of drugs, so small molecules. So we have libraries of drugs, like thousands of them, and we can grow cells, so the human iPSC cells, into nerve cells, and then we can put these drugs on the cells and sort of screen them in a high throughput fashion. This is something that you can't really do in mice. So this is a really great system for being able to do that. And we're doing this in collaboration with a company called Rumi Scientific, who have developed a platform to being able to sort of grow the human cells. These are sort of, again, mini brains, but in a slightly more organized fashion. They look like this. And these are examples of two other neurodevelopmental disorders that are very related to FOXG1, so Fragile X and Rett syndrome. And they show that when they grow cells from Fragile X patients or Rett syndrome patients, they look very different to the normal situation. And so the idea is that can we put these thousands of drugs in there, and we're just going to measure, can we turn this back into this? So that's what their platform is about, and we're very excited about that. I should also mention that we're doing a parallel screen in zebrafish in the lab of Corinne Huart and King's College. And she's using, instead of this platform, she's using her zebrafish model, but it's the same idea, same concept. So I'm going to just, I was told I need to give you like take-home messages. So I think these are the most important things that we learned that I want you to just remember. So we now know that we can make improvements in mice. We have methods to fill the bucket. So this is sort of like the targeted approach, and we know how to dial it up. We know how to restore it. But we need to fine-tune these methods more to be able to target the right cells with the right dosage and to limit toxicity. And we are in the process of identifying other targets, so through this sort of blind approaches, but also more understanding of the biology. So on the last point, I'm just going to end on how important it is to carry on still trying to understand the biology more and invest in understanding the basic sort of building mechanisms in the cells so that they can actually inform therapeutic strategies. And I would like to thank you all, say thank you to all the scientists who are here on Thursday, who are so extremely collaborative and open and shared so much of their data and generated so much discussion. So thank you to all of you, and questions.
Thank you, Hourinaz. So I would love to invite our scientists, Sue, if you could come up, Fabian, Angels, and Tiago. I'm so excited you guys stayed. Would love to have you come up as well, Corinne and Shibi. And then you guys can ask questions and anyone in the panel could answer.
One more chair.
Does everyone have a chair? Okay, super. All right, it looks like we already have a question. Go ahead.
Oh. Pam has a question.
Yes. Thank you so much for making a very complicated subject a little easier, just a little bit. So just curious about the scientific community and the sharing of information, because I, from the little bit that I know, right, a lot of times people can get sort of protective of breakthroughs, and so that's sort of the first question. And are our only options the FDA? Like, is there a licensing medical board in Germany or France that we could also maybe get drugs legalized? Or
So I will answer the first question. So you're right that often scientific community is not too collaborative, and this not getting all the information together delays the science. And I'm so happy to tell you that that's not a problem in our FOXG1 research community here. So I think that's what I meant when I say FOXG1 families and FOXG1 kids made us better scientists. We know what we are working toward. So we know that we don't have time to have internal politics and claiming my own science. So these are our colleagues who I have communicated regularly. And FOXG1 Research Foundation made a commitment to connect us and made really collaborative environment as well. So we bring our own expertise to the table to discuss how we can collaborate. And I was amazed how much progress we have made just by talking to each other and learning from each other's progress. So that's not a problem.
Yeah, and you know, Pam, one of the criteria we have before funding a scientist is that they're willing to collaborate and sharing their assets, and we put that in our contracts as well. It's a core part of our belief system. And you know, on your second question, I know you're not on the panel, Flortji, but I would love if you could maybe help answer that with the regulatory and you know how we're thinking about European agencies, et cetera.
Maybe introduce yourself as well.
Yes, please.
Hi, my name is. I'm Floortje van Houten. I've been in market access and commercialization in pharmaceutical companies for 20 years, and I offered my help to the foundation. I suggested that in Europe, the FDA equivalent is EMA, and it's 26 countries in one go. Unfortunately, the UK left, so that's now a separate entity. But I suggested you try to talk to FDA and EMA at the same time, because what you don't want to end up with is that the EMA has certain requests, and then the FDA has other requests, and then there's PMDA in Japan as well, and they do collaborate, so maybe there's a way you can talk to them all together if you are smart about it and they want to collaborate, because at the same time, there's not a lot of patients on the world either to do thousands of trials with what they want. So yes, there is a way to talk to them, and it's EMA in Europe.
Thank you.
Hi, I also just wanted to add on to the part of doing these in like multiple countries or even like multiple continents. One of the things that you also need to be cognizant about is based on the therapy, the requirements from these regulatories are different. So they might be actually more difficult. So for example, in case of like a gene therapy or so, some of the regulatory requirements from EMA or so are more stringent than they are in, I mean, FDA itself is stringent, but then you actually have some of these other additions also. Along with that, there are also the manufacturing restrictions, and then the restrictions also where you are manufacturing, how it is being manufactured, how the drug is being shipped. So where the first clinical trials also, when you're trying to do, if they are multi-center, even within the same country, that generally may not happen. So it might be single center trial, just so that it is controlled. But then if you actually start adding the countries and then continents, that actually adds even more complications. So that's where you would see for quite a few of these first in-human therapies, they start at a particular center or in a particular country. But as was mentioned earlier, there are efforts side by side to actually engage into different regulatories so that once the first phase is complete, you don't actually restart at the end of it. You're actually doing it simultaneously.
Exactly.
Yeah, sorry, I always have questions.
No, we love it.
Two things. If I have a great neurologist who wants to join the team, is that possible? You know, like, is that your goal? Do you want more science? Okay, wonderful. And so I'll just bring this notepad and hand it. No, what I was also thinking, I know you were working on a standard of care package, and as a parent, I'm thinking, you know, quality of life is my concern for my child, and also assistance to you guys to help the next generation. So I have found it very difficult to do the biopsy thing. That has been very confusing. My neurologist said she'd help, and we're in different cities, and it just hasn't worked. So I would love to see in your standard of care package that, you know, we're giving to like a two-pronged approach. What are you giving to parents? And what are you giving to the scientists, and making sure, because parents are the go-between, like, we know that this stuff's happening, and how do we get those biopsies over to you guys, and how do we help the scientists, but then also ensuring that, so basically, do we know everything we need to know, and then do the doctors know everything they need to know? So I'm sure you're thinking that way, but I'm just thinking like, that's a gap for me in my experience.
Yeah, no, thank you for bringing that up. So the first point, yes, we would love more neurologists to join the team. I know, you know, Tim and Eric would love that too, so we're not giving every question to them. But we are trying to grow the team in all ways: parent volunteers, scientists, neurologists, etc. So it's fully expandable. So yes, would love that. And that is a great idea when we think about our standard of care. Also something that you can hand in terms of parents who want to help drive research forward with the biopsy, et cetera. And we know that there are parents who want to do the biopsy and it's difficult. So we are actually looking at how we can circumvent that and potentially even work with another lab other than Coriell. So definitely.
My daughter was diagnosed 5 years ago at the age of 4, and at the time I was told that a cure may be possible someday for a FOX that has a mutation but not a deletion. Can somebody speak to that?
Who'd like to take that?
I can try to answer. So I think the treatments that would be available in all these different diseases are based on the mutations that you actually have, right? So we actually come from the gene therapy translational projects for these particular diseases. And so if you actually, if you see that one of the ways the gene therapy works is the gene replacement therapy. Although it's a misnomer, you are not really replacing the gene, but you're actually giving back a functional copy. Now that is also dependent on how big the gene is. Can you actually fit that into a virus so that you can add it back? But so in a case of a deletion mutation, you would still be able to have some benefit if you are able to bring back an additional copy of the gene that is fully functional. There is no deletion on that particular copy itself. So the gene therapy type treatments would be possible for a mutation that actually has deletions. But again, as I mentioned, so if that is not the case, if you actually have a missense mutation or so, you can still utilize different types of treatment. So from the biological point of view, a mutation that or a disease that has a deletion mutation, a gene therapy could be a viable approach.
I can just add to that actually some of the other approaches that we're undertaking, such as ASO, is also a viable approach. So it just acts on the normal copy that the child has by dialing it up. So that will replace the missing copy.
In some ways, I've heard that a deletion could even be the easiest because we know we just have to add more FOXG1, right? There's no other complications. So yeah, there's a lot of things that are said to us as parents, and unfortunately, many of them are just not completely true.
One of the questions also is whether or not your deletion is just the gene, or whether you have a lot of other genes that are deleted as well.
Yep.
And so there can be complications if you have other genes that are missing that may also cause problems.
That's right.
So just targeting FOXG1 may not be sufficient, or it may make
Help.
a difference in life.
Yeah.
It may not fix everything.
Exactly. Go ahead.
With the outcomes of the research that you did, where the brain regrew, is that a permanent thing that'll stay that way? Do you guys monitor the mice models to make sure that that regrowth stayed?
Sue, do you want to take that?
So in mice, I will add the previous comment and then answer your question. So from day one, we are committed to cover all different type of mutations, or not every single mutation, but this spectrum of mutations. So that's why we have generated different mouse lines that represent frame shift mutation, deletions, duplications, missense mutations, and nonsense mutation. So the approach that we are taking is we are developing the therapy that might cover all different kinds of mutation, but we are also exploring for individual category of the mutation that can be tailored for that type of mutation. So we have to understand the biology underlying the patient pathology, and then when we have better understanding, it really facilitates the development of drug. And now whether mouse brains grow again, so the mice are born with many deficits, but FOXG1, as I alluded, continues to work after birth, at least in mice. So there is a limitation to recover the whole brain structure deficit that the mice are born with. We can do that, some of those, as Hourinaz showed, we can connect the two sides of the brain, but we don't necessarily make a brain to the normal size. Having said that, FOXG1 does a lot of things like brain inflammations and also myelination that Hourinaz showed. Like FOXG1 is necessary properly insulate the nerve cables. So all of those things we can target and then we can rescue. So growing brain is not just the only goal that we are aiming for. We want to support FOXG1 to do this job properly.
Yeah, and we've talked about the different mutations, but I don't want the takeaway to be that every mutation needs a different therapy. Hourinaz talked about that this comes down to the protein and the protein being expressed. And so by looking at these different mutations, one of the big things, and Hourinaz, I'd love for you to explain this a bit more, is the mutation leading to a gain of FOXG1 function or a loss of FOXG1 function? That's really where the difference in what type of therapy becomes important. So that's the overall reason for why we're studying each mutation, not because we think each mutation is going to need its own $10 million therapy. So Hourinaz, if you want to just explain the protein being the most important and the gain versus loss of function, because that is something you will start to hear more and more us talk about. Is it a gain of function, loss of function?
Yeah, so going back to that schematic that I showed about how DNA makes RNA, makes protein, really what at the end of the day, what we care about in cells is the protein. And there's not a simple correlation between the mutation, RNA, and protein, which is what we're finding. And so you can have the gene being mutated and faulty, but and the protein being missing, or you can end up with a different form of the protein, which can actually go on and cause sort of other abnormalities in the cell. Or you can even have increase in the levels of protein. So that's why we're studying each mutation in both cell lines, in human cell lines that comes from the patients directly, but also in the models that Sue has created to really be able to understand whether the mutations are behaving just as if you would just delete one copy of the gene, or if they're behaving as a sort of mutation that is taking on additional functions in the cells. And so the easiest way to go about it would be to just target either the RNA or the protein, but there might also be other downstream sort of pathways that we can actually target if we can identify what it is that it's acting on.
Great. We have a question.
Can I add to that, and then I will take your question. So I think that there would be universal therapy options for the loss of function kind, whether it would be loss of frame shift nonsense or just the deletion of the gene. As Hourinaz said, and at the end of the day, it's about the protein. So we might have some faulty FOXG1 protein, but by adding enough normal FOXG1, this protein might compete, and then having enough normal protein may win. So as Nasha said, I don't think it's necessary that we have to develop a specialized therapy for individual mutations. We are still testing that options because maybe combinations of different therapies give the best outcome. But I think there would be more universal therapy options that cover broad spectrum of FOXG1 syndrome.
Maybe I can also complete that because the changes in PLP1 that you have seen a few seconds ago, what Hourinaz didn't have time to show you is that we generated a mouse model that has one type of point mutation, and then we identified these changes in the so-called PLP1. But we wondered exactly the same. We said, is that unique to that very specific mutation or not? And we have some evidence that we didn't have time to show you that we at least studied 4 different mutations, and this was common across the 4 different ones. And I personally believe that it's probably across all mutations. Therefore, targeting PLP1 it might be beneficial to every type of mutation, actually.
Add just one quick thing. So as your earlier question was also like, is the change that you are seeing is permanent, or is it going to actually lose also? So for any type of the therapeutic inventions are done, or they're actually being tested, at least within the mouse models or so, what we do is a short-term study and a long-term study. Yeah. So it is not that you are looking at it only at one time point. I mean, of course, in order to get the early indications, like if I inject a mouse today, I want to know as soon as possible if it is working or not. So generally, your earlier time points that you would check would be the time point at which you would see some difference. But that doesn't mean that is the only time point you actually check in the study. So when these therapies go to the clinical or the regulatories, you have to show a long-term efficacy, which is what it is called, that the effect is still continuous. Yes. And at the same time, you show long-term safety, that while that is happening, you are not actually having any other side effects. And that is true to all the therapies that would actually be eventually make it to the clinic.
Thank you. We have a question here from Maddie from the UK.
Hi, thank you so much. I just wondered if you could explain a bit more about what time frames we're kind of working towards for the path for a cure. Thank you.
Great question. Who'd like to attack that? I can have a go. Well, as you saw, there are many different steps involved, and we don't, basically the last thing that we want to do is to skip any steps. And so right now we have We have methods that are looking really promising, but we need to further refine them, and we need to really make sure that they're safe before we actually are even able to get. There's lots of regulatory steps as well that we have to fulfill before we can even test them in a clinical trial. So I can't remember what the time frame was for the Angelman syndrome.
It's a great question at the clinical trial phase, because obviously, a mouse life and a human life it's not a complete correlation. We can get some sort of estimation of the time frame, but I can't share. There's some details I just can't share. But the Angelman trial was a later age group. And the idea is that at whatever age you are able to receive the therapy, you're going to see benefit. You will 100% see more benefit at an earlier time frame, because that is just brain development, right? The brain will get more so it can develop better early on. But what we're seeing from these other trials is that no matter what age the child is given treatment, there is change. And what I was amazed with when I asked so many of you yesterday, what are the top 3 things you would change? What blew my mind is your desires are so small. It was, hey, just a little bit more motor control so they can just stand up. And it's true, like for us, any small change is a big change. Our child is now potty trained. That's mind-blowing for us. So we're really hopeful with the time frame that whatever point there's going to be some benefit. Now, the FDA has, different regulatory agencies will have their own rules on what ages dependent on the drug, and so we'll have to attack that when we get to that stage. But hopefully that makes you feel better, Maddie.
Can I add something to that? Sorry. As you can see, I love to talk. I think the one more thing that I would add, just from the time frame from a conception to the clinic, from a preclinical time point of view also is, so you have a conception of a therapy, but in order to actually see whether the therapy is working or not, you need to have reliable model systems. So if all of this is done one after other, you are really waiting for, you can't have a therapy if you don't have a model system to test for it. Once that actually is done, you still have to figure out, as I mentioned, the long-term efficacy, long-term safety. Once that is done, consider this is actually all done using a research-grade material. You can't use the same material for clinic. So now you have to make the clinical material. That requires additional testing. So all of these things need to get done even before you actually go to FDA in that case to say, okay, we think we are ready. Now, can you tell us what else is needed? And then you get that feedback, and then you most likely have to do additional things to satisfy FDA. The main thing over there is that it is safe, and there is a considerable optimism that it would be beneficial. And so then you go into the clinical trial. So all of these things take a lot of time. I think the FOXG1 Research Foundation has done amazing job, because if a single lab is trying to do that, it could take years. But when you actually have a collaborative work like this, it can be shortened, but it's not just within months.
Corinne, did you want to add something?
Yeah, I just want to add that I think it's a really impossible question to answer, in the sense that we really don't know what will work how. So in the sense that, we're trying complicated things like you just heard, or repurposing drugs that are already FDA approved. And so if we find a drug that's FDA approved, of course it's going to be faster than, but it's not going to fix everything in your kid. So, basically we could possibly hope that we will find something that fixes some aspect of the issues of your child relatively quickly if we're lucky with the drugs. But we don't know if we, see what I'm getting at. So it could be short or long, and it's impossible to answer. And I think that's the right, we're doing our best. And I want to reinforce the message of collaboration. When I joined the group, I really saw, first of all, for the first time in my life as a scientist, I saw a direct purpose to what I'm doing, which is amazing. Mm-hmm. And then that made me really realize that there's no doubt that it's about trust, and we can just share everything. And so everything that I've shared with you guys 2 days ago, was not published at all.
Yeah.
And with Sue, I have constant, interaction, because I think that, as soon as I found something that I find exciting, it's much better to share immediately. So we're sharing immediately, we're doing our best, but I don't think we can actually put the time to cure at all.
Yeah, maybe I don't like speaking, but I'm still going to do it. I think your question is not so much how long it's going to take, because no one in their right mind is going to answer you and give you a number. But I think that the real question is, how can you guys help us and how can we help you? I think you underestimate the way you help us. Someone was mentioning mosaicism this morning. That's something I'm also really interested in. And I talked to this person a few minutes ago. And that really fast track, you've seen that example with Angelman syndrome this morning. So if the idea is to go faster, I think that's the way and that's what the FOXG1, the FRF is doing so well, is bringing the community, not only the scientists on their side or the family on the other hand. And I gather that it probably is very difficult to get skin sample, for sure. But I can tell you it's really valuable when we receive it. We are on the receiving end, and that fast track what you've seen here. So as fast as possible is probably the answer.
Yep.
Okay.
Hi, I'm Kate from the UK. Unfortunately, my question's a little bit more pessimistic. I was wondering, with the mice and the zebrafish, did you see any negative effects after, the corpus callosum was regenerated? Obviously, presumably that's producing a lot more brain tissue all of a sudden. Were these adult fish and adult mice? How did that affect them? Did they suddenly have a huge amount more brain tissue crammed into the same size skull? Did that not cause other issues? How does a brain react when it's been wired in a certain way for a long time and all of a sudden something gets switched back on? Does that not cause other effects for different things?
So I will answer the question and have other scientists answer as well. So that's a wonderful question, and that's why we have to be careful not to have any side effect. In terms of specific gene therapy project that we have, when we are administrating these gene therapy options to our mouse model, at the same time, Shibi, Catherine Meyer's lab are injecting that to the wild-type mice to see whether there's any long-lasting adverse effect. So we didn't find that yet, but that doesn't mean that this is the safest gene therapy options. So we are continuously working to make it better, and that's why we have to, and we are not waiting to see whether there's any problem or not. We are doing everything at the same time while we are working these gene therapy options that we already have in hand. We are also at the same time developing a more better targeted gene therapy for FOXG1. So before these things go on to any type of human trials, we will make sure that there's no toxicity, and we are required to do that. Yeah. And then make sure that there's no adverse effect on cellular or animal systems that we have. And we are perfectly, all the scientists are aware that we should avoid all those things. And I think that at the end of the day, we will be able to find the solution without getting other cells stressed out or something like that.
I just want to reinforce that. So when we look at our models and we do something to it that seems promising, we don't look only at what we need to fix in the patient. We look at the brain overall, non-biased, and we ask, is this brain looking normal or not, looking at very different, a whole set of criteria. So we are very careful about what's happening and what's not happening, and it's a very important aspect of the study. What I want to add is I wouldn't want everybody to be too obsessed by brain size, because we know for a fact that you can have a bigger or smaller brain than average and still be completely normal in behavior and ways of interacting. So, you know, it is very possible that we would never, you know, recover. I mean, I would hope that actually the brain will not be increasing in size because, as you said, there's a finite skull, and yet work much better. So the corpus callosum is a cable that links the left-right to the right side of your brain. It doesn't really mean that you make more neurons. It just means that the neurons are now connecting from left to right in a normal way. It doesn't add information. So what we're speaking about is using the same number of neurons, the same kind of number of cells you already have in your kid's brain, but making them talk to each other in a much more functional way. So don't worry about brain size, I would say.
Yeah, and I think one of, Hourinaz didn't have time to show you all the data, but the other exciting breakthrough that we've seen is that we saw improvement in memory and cognition. And so that is something, obviously, that is going to, be close to our hearts of what we want improvement in. I know there's a question online. Go ahead, Ryan.
Yeah, a question from Rachel attending virtually. I'll just read it verbatim. So what do we know about how 14q12 deletions proximal to FOXG1 might affect FOXG1 regulation? Our daughter has a deletion right next to FOXG1, but presents with FOXG1 phenotypes.
So I would love to have that information, what region of her, Rachel's daughter's FOXG1 neighboring areas are deleted. So these are kind of information that will really facilitate the scientific advance. So we presented the data in Science Symposium. So we are looking into what area of the genome close to FOXG1 is important to boost to FOXG1's expression. So although you still have intact FOXG1 gene, if you're missing this piece of chromatin area, that's critical or that's necessary to boost the FOXG1 gene, you would have FOXG1 syndrome because there's not enough FOXG1 levels again. And having this human genetics information, what area is missing in that case, not to have enough FOXG1 level, that's really critical information because then we know that that area is really critical to increase the FOXG1 level. And what also one other way to translate that information is that when we are devising gene therapy options, we want to drive FOXG1 into the right place, and that area that Rachel's daughter is missing could be that area that we have to put into the gene therapy options. So this is what I meant and what all of us meant, that we really rely on you guys providing us with your child information, and that feedback us with what we have to do to make the advance, and then we can give that back to you to have you better understood and then have you better treatment options. So it's truly community. It's not just scientists and then parents, like separate two areas. I think we should talk all the time and then learn from each other.
Also related to this, I think will be, as someone mentioned, will be great to have these skin biopsies from these samples. We work in the human model. It's in vitro, it's in a dish, it's a very simplified reductionist model to study human brain, but it complements all these other researchers that can study the brain in mouse, like a physiological brain, but the advantage of the human model system is that we get all the genomic information from that patient is maintained in the model. So we can ask all these different questions at the cellular level that cannot be done in other models.
Maybe I'm going to do that again because you have a terrible influence on me. So I'm going to take an actual example to complete what you just said. So I actually came across a report from a family. The family had multiple tests, as you've seen. I'm talking about genetic tests, and nothing came up. And she had the family, had the typical, what you all most likely had, which is called the exome sequencing. Exons are part of the gene, but they're not the entire gene. And nowadays, although this remains the main way to detect this sort of monogenic disorder, we have the capacity to do what's called whole genome sequencing. And it turned out that for that specific family, the mutation was actually in the promoter or in the regulatory element. Therefore, it has only been detected when they went through I think it's called the Genome 1000, when they had the whole genome sequencing results. And I suspect that must be the case for other families where they're simply not detected. So another way where you can help is by lobbying whoever you know at the time where you can do now whole genome sequencing for genealogy purposes. If you can lobby the powers that be, whoever that is, so that you get actually whole genome sequencing that covers the entire genome of your kids, then that brings more information to us ultimately. So I think that the question was really interesting.
Just add one more thing. I think this is just another example of like how Sue also mentioned that the patient community actually helps. So within our lab, we also work on some of the in vitro disease modeling, which is again similar way as it's been actually done from patient fibroblasts to disease-relevant cells. And with that particular help from the patient community, one of the example would be SMART1 or CMT2S, where you actually have the mutations that cause the disease, but you also have some mutations that are the variants of unknown significances. So it's more or less you don't know what is actually happening. It might not be within the same gene or so, but in vitro, you can actually study those. So you can't really find mouse models for that, but when you have in vitro labs also that are working on it, you can utilize the similar therapeutics in the patient cells directly, which otherwise wouldn't be possible because you wouldn't be able to create a mouse model. So like all this work finally complements that it's not only gonna help one set of patients or so, it actually then opens it up globally.
I think we have another question.
Leah has a question.
I have so many questions, but the one that I think is most relevant to everyone is where is this a proper FOXG1 gene coming from that you're putting into the body? Is it from the human being you're putting, like where is it coming from?
That's a good question.
Yeah, it is a human gene. So thanks to the advances in the Human Genome Project or so, we do know what is the actual coding sequence of the human gene that will actually make the human FOXG1 protein. So that's where the sequence is coming from. Generally, in case of these gene therapies or so, you can now commercially synthesize these genes to make sure that there are no other mutations or anything. So you wouldn't take it from, you won't take it from a parent's copy, replicate it from there. You're really making sure what you're putting in is the exact thing that you actually want, but it is the human sequence.
Sorry, I don't really know who would answer this question, but it went back to the corpus callosum. So our kid he's almost 5 years old, and he already has like a thinning of the corpus callosum. Does that continue to be degenerative in FOXG1? And then how is that treated? So is that just the same thing? Like once it's there, it doesn't ever change until you can add back to it? Or does it continue to degenerate?
So when we made the mouse models for different type of FOXG1 mutations to cover the spectrum, most of them have corpus callosum agenesis. And we traced the mice for almost 2 years, which is the lifetime of the mice, similar to humans like 80s, 90s. We did not find any evidence of neuronal degeneration, or at least any remarkable neuronal degeneration. So I think FOXG1 is less of, or maybe not, neurodegenerative disease. This may be more neurodevelopmental disease. Now, can we restore FOXG1's corpus callosum problem? Can we reconnect them? That might be possible, and that's what mouse data suggests. But having said that, I don't think that's the most important part that we have to restore. So we have tested the mouse model that's treated with gene therapy, and these mice didn't have restored the corpus callosum, but they still show the improvement in cognition. So the corpus callosum, although we want to have it, but that's not the most or the only connections within the brain. There are a lot of other things, and a lot of other things that FOXG1 does, I think is important. So this is not the major or the only thing that we are aiming to recover. And even without recovering that part, we do see the difference in mouse's behavior.
And Nadia, it would be great for you to.
Yeah, I have two things to say. First, we don't have any evidence from clinic that it's a neurodegenerative process, so I don't think. And for the MRI, up to my knowledge, there's no degeneration. But we don't have natural history of the MRI, so maybe with the collection of the MRI that we will do with the registry, I will be able to tell you whether the corpus callosum is growing. And last question, we know patients without corpus callosum agenesis in our clinic, and they are functioning pretty well. So, as you said, it's not necessary. We will need to improve synaptogenesis, myelination, but making the corpus callosum, when it's absent, making it being here, it's not easy, and maybe it's not necessary.
Exactly. When I present my data, oh, we restored the corpus callosum, and clinicians will say, well, you know that's not that important. They are walking humans and functioning normally without that much corpus callosum, so what that means. So it's more important to find other things recovered.
And I just want to add quickly, real quick, is that the scientists here are looking for a marker to show that the therapeutic is working. Right, not necessarily that is going to happen in the patient or needs to happen in the patient. It's just giving them proof that what they're doing is having a positive impact, so that they're on the right track and continue to do that.
So from a parent perspective, because this was like, you know, we've known about this for like 2 years, this diagnosis, and just my own curiosity of how the brain works, I started researching like how to build neurons and how to build, you know, all of these things. So we do a lot of repetition. And, you know, like from a parent perspective, you know, can we see, I don't know how simple this is going to sound, but can we see growth or milestone achievement just based on our own repetition and our own therapeutics that is not from a medicine standpoint or a treatment standpoint, you know, from pharmacology, but, you know, is that as if, I don't want to say as effective, because I know that, you know, gene therapies and all of that are going to be the most therapeutic, but are we having a realistic expectation that there are things that we can do just holistically and repetition and introducing those things over and over and over, that we're still going to see an impact in growth in our child?
I know you're all scientists, but
yeah, I'll have to answer this question from the perspective of mother of Fox child, not so much from perspective of science. I think the repetition and giving the best care that's available to our children is very good idea. I'm someone who's studying the developmental biology of the brain, and there are enriched scientific evidence that the mice giving a better environment, more toys, for instance, really strengthen their brain function and make the mouse remember better. And that's one thing that we can try in FOXG1 mouse models as well. I think that will make a difference just based on what, based on the outcome that's already available. So I think environmental enrichment or continuous therapies will make our children do better cognitively and physically, motor control-wise. And I do see that anecdotally with my daughter. So that's, I mean, we are not scientists. We are not suggesting that, like, stop what you're doing. We are giving you something better. Everything has to be tried. And we are attempting, or we want to add more options, or the options that solve at the more core of your children's disease, but that doesn't mean that other things are not important.
And that's something our neurologists in the house might talk about after lunch.
Yep.
Shibi, maybe I know we discussed a little bit about some trials that were conducted at your center that children had varying outcomes based on their environments. So maybe you want to talk a little bit about that.
Yeah, I can definitely add. Again, I think even from any of the therapeutic development perspective, I don't think any of us would actually say that this is going to cure everything. There is a very realistic outcome that it can either rescue or stabilize. And so that also depends upon what the treatment is, when it is given, and what is actually wrong to begin with. So in case of, a quick example would be if it's a neurodegenerative disease where there is a loss of neurons, if you go too late, you don't have anything to actually treat. So that becomes a difference than when there is actually, the cells are still there. But again, being that said, the brain is still very plastic, especially the human brain all throughout. And all these other conditionings that are happening do have an effect on what the child's health is going to be or the quality of life is going to be. So we have seen in certain examples where even if you actually have given the therapy, the environment that the child is in does really affect how good the therapy is going to actually perform. So in case of some of our Batten trials also, the patients actually have speech impairments, vision impairments also. But even when the gene therapy is given, we continue with their speech therapy. We continue engaging those in the other ways. So it's not really that that's your silver bullet, that if you have this, then you don't have to do anything else. All these other things that are there continue to help improve the quality of the life. And they help then the therapy developers, because now you have better outcomes that we can actually measure and actually show that it's working.
Okay, are there any other questions from the audience? All right, I'll have a one wrap-up question and then we're gonna head to lunch. All of you are working in different areas and you have different specialties and you also work on other conditions. I'd love just if you can give some insight on how far, and I think this is a big question for parents, how far along do you believe we are? Do you believe, honestly, that clinical trials are in the near future? Or do you think there's just so much work left to do?
Should I start? Okay,
Good,
sorry
Shibi.
again.
And I just want to also point out that Shibi's team has developed Zolgensma, which is a gene therapy that is on the market, passed through clinical trials. Children are taking it. So Shibi was part of that team.
For SMA.
Yep.
For SMA. And it's a big reason why we chose to work with the nationwide team, because truly that is one of the most, if not the most successful gene therapy. There's lots of scientists making gene therapies, but to develop something that is actually effective in a human being, not just an animal, does take a different type of rigor. So with that, Shibi.
Yeah. So thank you for that introduction. Also, I think I'll just quickly add. So I came here in 2009 for to do my PhD with Dr. Brian Kaspar. And then within last 13 years, I have been really just fortunate enough to be part of multiple gene therapy trials from Nationwide Children's, Zolgensma being one, which is the intravenous. So you're actually giving the therapy through blood. But what we have also actually developed throughout last couple of years is a different route of delivery, with which now we actually have 3 clinical trials for Batten disease and then also SMART1 in the clinic. So considering how much time and how much efforts had gone in getting these trials from conception to the clinic, I definitely see that we are in a much better position when it comes to the FOXG1. Again, this is being cautiously optimistic. In order to get this towards the finish line, there is still a lot that is actually left. But what has been most amazing is the collaborative work that actually makes it fast track it, rather than trying to do it just by one person or a single lab. So yes, from our perspective, yes, we are very cautiously optimistic to get this, and we will make sure that we see it through the finish line.
Yeah, and I will say that everyone, and there's over, supposedly online, it says there's 7,000, but we now believe that there's over 11,000 rare diseases, and I believe 10,000 of them have written to Nationwide to work on their gene. So we're really thrilled also that they've chosen FOXG1 as a main gene. So, any other comments? All right, should we rise? Everyone hungry? Okay. All right, Nicole.
Yeah. Okay, so that's half of the day. We have lunch served outside, and go enjoy.
What time are we back, Nicole?
We'll be back in 1 hour, so 2
Okay.
o'clock.
2 o'clock,
few
all right.
minutes
And
before
thank
2.
you guys so much.
Yeah, thank you to this panel.